This is Tiffany Tulao, week 2, lab experiment 1 of week 2. It's aseptic technique in culturing microbes. I have all my supplies here ready and I will go over them. To get started for this week, I have laid out all the supplies that I'm going to need for this experiment, and I have them all ready to go. As you can see, I have my pre-poured plates of agar that I made ahead of time so that I can start this on time. First and foremost, I'm going to start out by washing my hands properly to begin my lab. As you can see, I have applied my gloves, safety goggles, mask, and apron to begin this lab. I am now going to disinfect the work area with bleach. You see, bleach. This is my waste bucket for disinfection, undiluted bleach, or for any waste to treat anything. This is the cup that I will be labeling as alcohol, and I will be pouring it half full. It's about half alcohol. And I will be placing the pipette so that the stem enters the tip. I'm now going to label a 5 ml nutrient broth tube as E. coli. You can see. And I'm going to light the candle wick. It's kind of hard to do with gloves on. The lighter. I'm going to expel and sterilize my pipette to ensure that there's no more alcohol inside. You can see here. Now we know it's sterile. To relight my candle. I'm going to remove the cap from my nutrient broth and run it through the flame to sterilize the end of it. Still shaking my pipette to make sure it's sterile. Repeating steps 13 and 14. I'm going to do the same for the E. coli culture vial so that I can sterilize the end of that as well. I am now going to pipette 2.5 milliliters of broth into the E. coli container, being careful not to touch the rim of the sterilized specimen container. Still holding the pipette, I'm going to replace the lid of the E. coli container to shake and dissolve E. coli pellet, which it's dissolved. Now that it's dissolved, I'm going to transfer all of the specimen from the culture vial into the broth. So I've transferred it. Now I'm going to repeat the step for sterilizing the end of the broth tube before I replace the lid. And I'm going to stick the pipette that was used into the cup of alcohol. Now replace the lid. I'm now going to repeat steps 9 through 20 to do the same thing for this Staphylococcus epidermis. I'm going to sterilize the nutrient broth for the Staphylococcus epidermis. As well as the pill. It's plastic, so I do have to be careful not to melt it. And I will now take the nutrient broth and add it to here. Now shaking the vial to make sure that the capsule disappears, and then I will transfer it back into the vial of broth. I am now taking and retransferring the Staphylococcus epidermis into its nutrient broth vial. The capsule has completely dissolved. Get every little bit of it. Mixing in alcohol. And I'm now going to sterilize the end again so that I can put the cap back on. I'm now going to be adding a half a teaspoon of the yeast to the disposable cup. Setting this aside for later. And then I will be adding a 1/4 cup of warm tap water to the yeast. And I'm going to swirl this until it is dissolved. And I'm gonna let it sit for 10 minutes. Now going to transfer, well, first sterilize the broth tube for the S. cerevisiae. I'm going to pipette, after my pipette has been sterilized already, 2.5 mils of the yeast mixture into the broth. Sterilize once again. And replace the cap. I've disposed of everything and I'm getting ready to wipe down my counter space. My tubes to incubate in the place I discussed in the beginning of week 1 to incubate for 48 hours. And now I am going to take a closer look and take some photographs of my growth on my plates and then complete my video.